Okay, I hope you can see my screen and you can hear me well. If everything is not okay, please let me know. So, hi again. Today I will be speaking about our effort to build a COVID-19 disease map. So, basically, a repository, a computational repository of SARS-CoV-2 virus host interaction mechanisms. I'm from the University of Luxembourg, but I will be speaking about on behalf of the entire community that helped to contribute to building this resource. But before I jump straight into the map itself, perhaps to have a broader picture in mind, in the past years, and actually past decades, we're learning more and more about the complexity of the symptoms and mechanisms of various diseases, and the dream of the Human Genome Project that there will be one gene, one disease mapping is definitely not true, and we are learning about more and more complexities of how different factors play together. And at the same time, we learn more and more about our physiology and pathophysiology from different sources and from a growing number of tools that allows us to interrogate different layers, different scales of our well-being. And integrating and interpreting this data, this complex data sets, is very challenging. But the demand for this interpretation is actually growing. So if we'll take a look at the discovery in systems biomedicine, following discovery in systems biology, as illustrated by Hiroaki Kitano in his paper in Science already 20 years ago, we are starting with existing knowledge about an issue, and from there we are starting our data and hypothesis-driven modeling, leading us to certain predictions, which are then validated in wet lab or in real world, as you would say, which then the acquired delta then helps us to revise our knowledge, perhaps find contradictory issues and improve the modeling, and hence the entire process repeats. And already on the second step, you are going to see that this is a data and hypothesis driven. So as I was mentioning earlier, we have a lot of data sets flowing to us from different sources, but usually what is needed is not a data table that we are being fed with values and associated p-values, but we would like to interpret these data tables to gain insight into what do they mean. And this interpretation can happen on different levels, on the level of molecular signatures, so groups of genes associated with a given process, like you would be doing by overrepresentation analysis in gene ontology or GSEA and MSigDB, which gives you an association with a process or pathway. You could take a look into a more granular, fine-grained knowledge by taking a look at network repositories, which tell you a little bit more about interactions between molecules, like OmniPath, Intact. Or if we are interested in a more graphical and detailed narrative of what is the sequence of events, we would go for pathway databases, like in case of WikiPath with Reactome or KEGG, for diagrammatic representation, but a more detailed and mechanistic narrative about that. And in that vein, disease maps are a paradigm where the systems biology or systems biomedicine diagrams are being constructed in the standardized way with a primary focus on disease mechanisms. And not to take anything away from the approaches I mentioned earlier, molecular signature databases are more and more often introduce disease-related signatures like MSigDB, hallmarks of cancer, and so on and so forth. Network repositories start to getting geared towards diseases, like senior database or dedicated networks in Index and so on. Pathway databases like WikiPathways and like Reactome also introduce new dedicated diagrams for disease mechanisms. So in general, all across the field, we are seeing a growing demand of contextualized information about given disorders. And people more and more realize that looking at the canonical representation of mechanisms does not cut it. that we need to have a more focused, a more narrow representation of mechanisms which corresponds to a particular disorder. And this is where comes disease maps with this mindset. Repositories are being constructed to reflect solely mechanisms governing or implicated in a given pathophysiology. And disease maps put an emphasis on bridging the communication gap between the bioinformatic experts and between the domain experts like clinical researchers or life scientists. In that sense, that by quite verbal or by direct graphical representation of the context or replicated processes, disease maps offer a nice entry point into a complex world of networks or systems biology mechanisms. And the disease maps at the same time at the very bottom are resources which are quite well structured and standardized. So this type of a dual approach allows us to navigate and explore existing knowledge in a understandable fashion, but at the same time use this knowledge into more computational tasks like visualization of omics data sets, or by pulling down the contents of the diagrams to analyze them from a much more computational angle. So basically, to summarize the introduction about the disease maps are conceptual models of disease, which are meant for knowledge-driven data interpretation and modeling, with an emphasis on standardized encoding of the contents, intuitiveness of exploration, so basically a visual appeal to what is being explored, the possibility of more advanced visualization of omics, and computational endpoints for reproducible exploration and analysis. And I encourage you to check out diseasemaps.org to basically learn a little bit more about the entire concept. So almost 2 years ago, when COVID-19 became a global threat, a global pandemic, we started to reach out, and I guess everyone did, through our scientific networks, to see who would like to contribute to a project like a COVID-19 disease map. And in parallel, I really need to say that I'm feeling proud as a scientist in general, as a researcher, seeing the tremendous response of the entire scientific community to help decipher this new infectious disease. This was a global effort, and our particular part was to try to work out what is known about the molecular mechanisms of COVID and how they are being encoded. The whole plan was to go through whatever is known from the point of view of literature, integrate it into standards, and combine it with perhaps additional existing sources to create a repository for exploration, knowledge exploration and modeling, which then can be used to produce new scientific results, which then are going to be feeding into the further steps of review and curation. And this very much follows the logic of the discovery cycle in systems biomedicine I mentioned earlier by Kitano. So the response for the COVID-19 disease map project was tremendous. Over 250 people said that they would like to contribute in one way or the other. And the expertise that we tapped into was very, very broad, starting from clinical researchers through life scientists, through data scientists, computational biologists to bioinformaticians, from academia, pharma, bioinformatic SMEs. So this group was very interdisciplinary, with a very broad expertise, and quickly started to set up communication channels and so on in order to basically to organize ourselves a little bit. And here we relied quite a lot on, you could say, flexible communication or flexible documenting platforms, and also another key to the effort was having immediately regular telephone conferences, basically to organize ourselves. Organizing TCs was not that difficult at that time. Everyone was locked down, everyone was at home, but still people really wanted to participate, really wanted to contribute, and this is really remarkable. So the entire project on the COVID-19 disease map focuses on three main efforts, or I would like to illustrate it or discuss it from these three main areas of focus. So first would be the curation and content sharing to ensure that the diagrams which we are going to encode, we are going to build, the maps are going to be standardized, that they are going to be used, and that these parallel efforts that I'm going to speak a little bit in a moment, are going to be well coordinated. We also worked on integration and interoperability, such that the content which is being created and perhaps additional resources available outside are can go together, can be combined for analytical workflows and can be used for downstream analysis. And we also started to investigate whether we can use interaction database and text mining to support the curation effort, because prioritizing the literature is not an easy task, and whoever worked with systems biology or systems biomedicine diagram building will know that the curation time or model building time is the biggest bottleneck of them all. And last but not least, the integration and interoperability access was to provide easy access to the curated content that we are bringing, not only among ourselves, but to the entire research community. And the last point into this effort was the analysis and modeling. So we very early on started to work analytically on the contents that were built, which was even for prediction and hypothesis generation, which allowed us to very quickly spot quality issues, workflow issues, but also and feed them back to curators, such that the diagrams which are being built were built in an analysis-friendly or modeling-friendly way. So the entire effort combined into a really rich ecosystem involving curators, domain experts, and analysts and modelers. And although this figure might be a little bit complex and a little bit too much, I will do my best to try to guide you through it piece by piece. So let's start with the curation and content sharing. So for this, we were simultaneously developing systems biology diagrams, and here the curators were organized. So basically, we were privileged to work with curators from two major pathway initiatives, so Reactome and Wiki Pathways, and to have a large group of independent curators using, could say, standalone editors to provide the efforts. So let me emphasize this once more. We had pathway databases as participants, or basically curators from pathway databases participating in the effort, and people who joined in laterally and with their experience, or bigger or smaller, with Cell Designer or other diagram editors, were also producing diagrams. So in this case, basically, at the moment of my presentation, the independent diagrams submitted to our GitLab repo, we have 22 diagrams submitted. Over the Wiki Pathways, there are 32 diagrams available right now, and Reactome Pathway database involves two big diagrams of SARS-CoV-1 and SARS-CoV-2 replication cycles. So from that point of view, it was quite challenging to actually reconcile this, especially that Wiki Pathways and Reactome have their own visualization interfaces. We needed to, for the independent diagrams, we needed to store them, to version them on GitLab, because this was basically the common denominator we found. And we used the Minerva platform, a tool we are developing, to visualize them in a consistent way. So it was very interesting to work in this kind of a broad curation project involving that many different efforts. So Crowdsourcing this biocuration was quite a challenging task because at the starting point, we had these 3 parallel curation efforts, Wikipathways, Reactome, independent diagrams. The participants on the side of Wikipathways and independent curators had mixed backgrounds in curation. Not everyone was an experienced curator. People really wanted to contribute, but sometimes they had less experience in drawing the diagrams. The work was urgent, but this viral infection was quite new. And this is quite of a challenge, because while building disease maps or disease-related pathways, the first thing one does is they consult the domain knowledge. What are the key pathways being affected by the disease, and so on. We didn't have that for SARS-CoV-2. We had a lot known about SARS-CoV-1. There were other coronaviruses, but basically when organizing this curation, we had this strip of knowledge I'm showing you, so the paper by Gordon and al. At that moment of time, it wasn't even a Nature publication, it was a bioRxiv preprint. So we had this set of small sub-networks from the interactome and a very, very good and thorough review by Fung and Liu on human coronaviruses. So from that point of view, that was kind of our 2 beacons along which we were navigating ourselves. The way we did it was we started by reaching out to experts to guide us a little bit on what makes sense on coronaviruses and the curation and so on. We started to partition the curation content among different teams that would like to undergo, they would like to focus on some subsets of the Gordon paper, or perhaps follow coronavirus-associated pathways following their expertise. In order to help the curators to harmonize the content, we came up with common guidelines, such that we more or less know what is the common denominator of the maps we are going to develop, and we quickly followed up by regular TCs to coordinate. The work was versioned, and we followed systems biology diagrams and systems biology standards, SBGN, SBML for graphical and data level encoding. And here a big shout out to Verdam Hub, people who really helped us to quickly set up a project on Verdam Hub, provided us with extra functionalities, and this allowed us to immediately be transparent about who contributes to the project, who's working there, who's engaged. So, and just to show you a little bit of the challenges of the diagram building of how this curation was challenging, was that, let's for instance take an apoptosis diagram curated by Vidisha Singh, and if we would like to translate it to different tools which could be used to build these diagrams in different formats. So Cell Designer original graph is top left, and here there are possible representations in SBGN, SBML with layout render, or in PathVisio. You're going to see that some of the general idea of a layout is preserved. There are some quirks. I will move to that in a moment. But the fact that we had in our group people working with so many different standards helped us to actually improve the translation procedures. And right now, the translation across these diagrams is even more efficient than this one. So if you take a look at a really minute details, you will see that some of the edges are missing, some of the graphical representations are not there, but still overall the layout is being preserved, and we are working on figuring these things out. And this is important in crowdsourcing projects specifically, because first of all, you would like to let people know that they can work with the tools that they are comfortable with, and then that the formats that they're going to produce are going to be then harmonized. So to summarize the curation challenges and the curation and sharing of the diagrams, the goal is still simultaneous development of systems biology diagrams. At this moment, we harmonize diagrams between independent curators, Reactome and Wikipathways. Standards and guidelines, adhering to them was key to have content which is harmonized. And we were reviewing the diagrams using something called review checklist. This is a simple underestimated tool which allowed us to very quickly where there were no automatic procedures for content checking to review the diagrams in a harmonized and uniform way. What we have is, as a challenges, is that we still face tool and format specificities. For instance, I think there is still no good representation for a unified representation for protein complexes across different diagram editors, which we deal with. Curators still face a lot of difficulty to annotate the diagrams that they draw. And this is important to have the provenance of the mechanisms which have been built. And the content which is being built is multi-scale and multi-context. So it's difficult to combine and it's difficult to cross-check. So just to give you an example, an FKB activation appears across different diagrams in the resource, and it's being slightly differently represented across different instances. And yeah, basically this is a challenge. I think it might be a good point to, if there are any questions at this moment of time, I would be happy to take them or answer them. And if not, I will move ahead and we can also have a Q&A towards the end of the presentation.
So I'm checking now the section with Q&A, and for the moment there are no questions here. Actually, I had one about the challenges, but you already addressed that one. So,
yeah. Okay. So we
perhaps
it's fine. If there are no questions at this moment, we can move ahead. Okay. So let's move ahead to integration and interoperability. So another part of the diagram that I was showing initially. So the goal was to access structured, annotated, and harmonized content. We are building these diagrams across 3 different efforts, and it would be good to access them or basically combine them in a harmonized way. I mentioned that these diagrams are quite well translated across these different formats, but we also would like to combine them with enrichment from the text mining and interaction databases to guide the curation process to complete the diagrams and so on. So for that, we worked on format interchange procedures. So between systems biology layout, translation of these diagrams into network format, and support for modeling. So translation of these diagrams into SBML, SBML qual. For the interaction databases, in order to suggest curation and complete network models, we reach out to text mining and yeah. And another part of the integration and interoperability was to have easy access to the curated content. The diagrams which were produced, especially by independent curators, were deposited on the GitLab, and this doesn't give you an easy and straightforward access to visualize them and to explore them. And of course, it would be good that these diagrams are accessible programmatically and if you have certain conversion scripts. So let me share my, I will continue sharing my screen. I will just give you a glance of how this integration works in practice. So this is the COVID-19 disease map website on the Minerva platform, where we basically put together independently curated diagrams together with the diagrams on Wikipathways and Reactome. The way it works is that we have this online diagram which is browsable. The engine is OpenLayer, similar to Google Maps. As I was mentioning about disease maps earlier, this gives you a nice graphical entry into the contents of the entire resource. But when you click, for instance, on a virus replication cycle, quite an obvious diagram curated by a lot of people, you will see that we have the diagrams from Reactome pathways, SARS-CoV-1, SARS-CoV-2 infection, and another diagram for Wikipathways available for the same. So from this, I can open a diagram which has been, sorry for this hiccup. Let me just refresh the resource, and we'll see. Okay, so this is the virus replication cycle, which has been drawn by Marcio Asensio, so one of the curator contributors. From here, you can zoom in and zoom out into the pathways, check the annotations of elements, check annotations of interactions, and basically this way, it's way easier to explore this than downloading the file or opening it in Cell Designer to check it yourself. On top of that, we have diagrams, 22 diagrams paired like that, these independent diagrams I mentioned. And if you are going to look for ACE2, for instance, you are going to see the results that show you that you have ACE2 is in renin-angiotensin pathway, virus replication cycle, ER stress, and coagulation pathway. You can see where they are, and you can basically open the associated diagram. So this allows you to cut through the or cut across the diagrams that are combined like that. And for the virus replication cycle itself, from here I can jump into the Reactome pathway browser to show how this diagram looks like in Reactome. And I can also jump to the wiki pathways to show the same diagram in the wiki pathways. Okay, it's not loading. Okay, there it is. So that's the virus replication cycle as curated in wiki pathways. So this way we are trying to combine to integrate within this platform that you see, Minerva. We would like to combine the independent diagrams into one resource that is browsable, and we would like to also combine across different resources, so together with the pathway databases. Okay, let me jump back to the presentation, which is here. Okay, so I hope I was able to pass on the message that we took these diagrams, which were developed by all these independent curators, and put them to work in one place. So this is one thing that was very nice to see and that we managed to do. But also what we managed to do is we combined inputs from different interaction databases and text mining resources to enrich these diagrams. Of course, in not a very straightforward way, but what we managed to do was for instance, this. So the solid border elements here are new potential targets or new potential regulators of pathways which are already available in COVID-19 disease map. So these come from text mining resource from Indra. I think Indra was being presented quite recently in this very format, and down-filtered by the contents of OmniPath DB. So we had a COVID-19 text mining data set, filtered for quality against an interaction database, and these new potential interactors, which were not yet in the map, are right now depicted here. So a curator sitting in front of the COVID-19 disease map looking for instance, to work on PAMP signaling, they can take a look at, for instance, some interferon-related receptors which regulate JAK, or some specific TLRs which interact with my D88, and so on and so forth. So the solid line elements are the ones which are new. The empty borderline elements are the ones which are in the map. So this way, from text mining and from interaction databases, we were able to grasp something that curators didn't yet introduce in the diagrams. So for the integration, for the progress and for the challenges of it, the goal of this is to access to structured, annotated, and harmonized content. And for the progress, we were really able to have programmatic access across a number of resources, which were able to be explored visually, and some dedicated translators were being built. For the challenges, we still don't have unified format for workflows for all these resources. Each of them have their own specific computational steps, but you are slightly different. You're slightly differently processing the content from, for instance, IMEX or Senior, because they are directed versus non-directed networks. Same you work with text mining versus known interaction databases, and so on and so forth. These resources have different identifier schemes. So sometimes you're dealing with AGNC and TRES versus UniProt. This is even easier, relatively easy, because you are always dealing with something which is on gene protein level. But when you try to bridge together with, I don't know, phenotype or drug, whatever related nomenclature, this starts to become quite a challenge. And visualization of diagrams which are constructed is relatively easy, but visualization of text mining or interaction databases contents is not so. Usually you end up with a really dense hairball of everything interacts with everything, and that's quite challenging to process for curators in order to make sense out of it and curate it out into a regular diagram. Okay, so this would close the part on the integration of the different resources within COVID-19 disease map. And if there are any questions, I'll be happy to answer them now.
I can't see any questions in the Q&A tab, but so perhaps we can keep a couple of questions as we end.
Okay, great. So let's continue. So, and last part is about analysis and modeling. As I was mentioning, the idea is to take the contents of the diagrams and interaction databases, if available, and provide them as an input for downstream analytical analytical and modeling workflows. The purpose of that is basically to allow quick assessment of the diagrams, whether they're useful. And when we started the project, actually a number of different groups joined, which had expertise in the various analytical and modeling pipelines, starting from network analysis through kinetic and Boolean modeling up to multi-scale modeling even. And all of these started to work with the content, giving us a glimpse into what these diagrams are useful for, and what are the translational steps which are needed for it. So just to give you a very quick summary of what could be done from the point of view of analysis and modeling with the map. And this is just a teaser, because at this moment I'm discussing the resource, not the analytical outcome of it. So one of the analysis endpoints is a simple visualization of omics data sets, which relies on the layout of the diagrams and their annotations to actually help to interpret omics data sets. And here I also would like to jump again back to the Minerva platform, because here when we were making the map public, we also provided a couple of publicly available, so a couple of data overlays for publicly available resources. So basically, if you are clicking one of this resource, the contents of the map are being colored following the profile of the transcriptome. So here you see more or less how this pathway is being differentially regulated. You can look inside for particular elements and their differential regulation. It's here, for instance. So you see differential regulation of these elements being portrayed. If you take a look at another data set, is going to be shown side by side in this pathway, and yet another one, it's also going to be shown side by side. So these are for 3 types of airway secretory cells, and basically you see that this NF-kappa B element is active or is upregulated in airway secretory cell type 1 and cell type 3, but not cell type 2. IRF3 is only upregulated in airway secretory cells 3. So this is for pump signaling, but with this you get very quick overview of which different pathways and how they are being differentially regulated in this. And funny enough, or interestingly enough, Every secretory cells 3 are the only ones which are getting infected by the virus. At least this is the conclusion from the publication associated with this data set. And you will see that in the virus replication cycle, you see this cell type being the only one upregulated. If you are going to go inside, you are going to see that the upregulated element is TMPRSS2, so one of the key factors governing the viral entry into the cell. So this type of visualization of omics gives you a much better feeling across different diagrams in detail and in general about the overall deregulation. Now let me jump back to the presentation. So as I was mentioning, visualization gives you this possibility of immediately interpreting even complex data sets. Mechanistic analysis, in this case, a Dorothea pipeline, allows to benefit from the layout of the diagrams, their connectivity and annotations, and omics datasets to provide new insights in a more detailed way than just putting colors on top of the diagrams. In this case, the Dorothea pipeline was used to figure out new transcription factors which might be regulating this core signaling part of the interferon signaling pathway. And last but not least, Boolean kinetic modeling benefits from the parameters, connectivity, and the annotations. Layout is way less important here, but using drug targets or combining with omics datasets, one can formulate a little bit more detailed predictions. Here, I'm just giving as a teaser is a first attempt in a PhysiBOS to emulate the behavior of different cell types or different cell populations, the graph at the bottom of the page, under different knockouts of, so each cell population carries inside a small Boolean model of apoptosis, and then by tweaking with different levels of knockout of particular key regulator elements, you are able to see the dynamics of each cellular populations. Again, this is a teaser. I'm not going to discuss the results per se here. It's just to give you a feeling that a well-annotated and well-constructed diagram can serve as a basis of a model. So to summarize, the goal of this analysis and modeling axis was to provide input for and develop analytical and modeling workflows on the basis of the diagrams inside of the COVID-19 disease map. And we've made a lot of progress here where there are decent API endpoints available for a range of workflows. We are already having first analytical results discussed with curators and domain experts, and this provided also feedback to curators to help with diagram development. There are still a lot of challenges. If you ever dealt with SBGN, most likely you faced the discrepancy between SBGN process description, activity flow. Basically, it's the granularity of different diagrams, how they are being drawn. As a matter of fact, so far, the analytical pipelines which were used the most intensively relied on the simpler formats, the simpler contents of the map. It's understandable. It's easier to use a network format or Boolean modeling approach, which do not require parameterization. But still, this is something that we are seeing at this moment. And the final challenge that I want to mention here is integration of models and modeling results, because the models have different physiological and temporal resolution. So combining the results and their outcomes is not such a straightforward thing to do. So all these efforts was to it's basically an aim to build a computational systems biology repository of COVID-19 mechanisms. We would like to, or we are aiming, striving for development of systems biology diagrams, coordinating among multiple curation efforts, to give access to structured annotated content, and provide input for analytical and modeling workflows. And this wouldn't be possible without the COVID-19 disease map community. This is a community effort. And besides the fact of people contributed contributing their heart and soul into the project. Some of the key aspects that helped to develop the project further, I believe, were that we were quite transparent. We tried our best to be transparent with communicating about our goals and contributions, and what would like to achieve and who does what. And of course, the hidden heroes are data and analysis standards, allowing to put all these building blocks together. So I hope that this picture right now is not as scary as it was at the very beginning. Just to recap, the complexity of the interplay between different groups participating and different contributors providing the resources and their expertise to make this happen. So to summarize, the COVID-19 disease map project is a community effort to encode and investigate mechanisms of COVID-19. We are bringing together different experts, and this interdisciplinary group is, I have to admit, it was a huge learning exercise for me to listen from all these different angles, from diagram building through format translation down to the analysis, to that this whole ecosystem has to be, or benefits greatly from having everyone in the boat. And we hope we are able to, or at least we're trying to our best, to build a transparent, reproducible content, not only for us, but for the entire research community. Feel free to check the effort, or if you're interested in contributing, feel free to reach out to us. And of course, I really would like to acknowledge a couple of groups, including the COVID-19 disease map community. So without these people, the project wouldn't exist. The Freedom Hub team, who helped us to very quickly establish this transparency platform, or transparent project on their platform. In particular, I want to reach out to the R3 team of LCSB, who at the early weeks of the project immediately helped us with setup of some key and critical infrastructure, and Elixir nodes from Elixir Europe organization supporting the project. And these and many more people contributing are co-authors of a paper that we published October last year. I encourage you to take a look. This paper lays out a little bit more in detail our work, our project that I've just described. Thank you very much. I will be happy to take questions.
Thank you very much, Marek, for this great talk. So we have a couple of questions now. I think we need to go in our order. So basically, there is a question from Anna. I'll read it. Thanks for a great talk, Marek. A couple of questions. A, what are the next steps of the community? And B, can machine learning models be integrated in the pipelines?
What are the next steps and machine learning? Okay, so the next steps. The project is ongoing. So at this moment, we are a little bit more mature, and we also have more knowledge than we had at the starting point. So we already started, and the next steps is to take a look at the diagrams which have been developed and try to harmonize them a little bit more. As I was mentioning earlier, we had this roadmap for curation, and but certain groups did a very good job at taking the Krogan paper and building diagrams around specific viral proteins. They mapped out pathway pieces and linked them to a particular viral protein. Right now, this content is ready actually to be repartitioned among pathways where they belong. So reorganizing the content of the map is one, and trying to go across the board again, perhaps with text mining results and interaction database results would be another step. And finally, seeing where we could have a sustainable curation efforts to continue developing of these diagrams. Initially, they were fueled by everyone contributing to it. Right now, people get back to their lives, and that's absolutely normal, which basically means that we are losing this fuel of diagram building. And whoever worked on a disease map or related project knows that curation is very difficult to fund. So basically, for us, it would be good to look for resources to ensure that this curation ever continues. Whether the machine learning can be incorporated in the effort, I think yes, it would be very interesting. I think partially it's already happening because some of the text mining solutions that we deal with have machine learning components. Perhaps analytical endpoints. I think that there are many possible entries into the project from the machine learning angle.
Okay, thank you. We'll move to the next question from Matthew. Has COVID-19 map been utilized in any recent publication?
That's a good question. I don't think so. I think we published recently, so October. We are planning the analytical paper, basically to show a little bit more in detail to the scientific community how these workflows can be used a little bit more. But if the map is being used for knowledge exploration, this is up for checking. I've seen that the paper on the COVID map has been cited a couple of times, but to be honest, I haven't been tracking whether it was on experimental publications or was it, state of the art, whatever part.
Mm-hmm. Thank you. We'll move to a question from Thomas. So great talk, thanks. Does the resource also accommodate variant-specific knowledge like the putative molecular biases of different responses of different virus variants to immune system and drug challenges, different invasion of target tissue, etc.?
Yeah, that's a great point. Both viral variation and host variation are great avenues for research. I think that, as with every variant-related knowledge, we are a little bit stuck on the mechanistic interpretation. So as long as you have a mechanistic interpretation of what a variant does to a virus, so if you have a quantification of whether it's stronger or weaker binds the receptor or something like that, like, for instance, the spike protein mutations and so on, then you could analytically visualize this in the map. The same would go if you are finding particular variants which are present in subgroups of people having susceptibility or having resistance against COVID. That would be interesting to just put these variants on the map, visualize which genes are being targeted. But interpretation of genomic information needs to happen on the mechanistic level as well for it to be introduced into the map. The map is primarily a mechanistic resource.
Okay, thank you. I also had a question. So I think you already mentioned a couple of them, but just to summarize a top 3 lessons learned from this project and how much a new disease map project can be accelerated by the efforts done in COVID-19 disease map. So kind of like in benefit in that sense. So top 3 lessons learned.
Oof. Top 3 lessons. I think that having like a full across the board integration of different players is key. So far, I was implicated in the disease map projects where we were building diagrams together with clinical researchers. And that was the scope of the disease map project. Now we had this component plus what are the external resources that can help this building, and we had the downstream of what are the analytical pipelines I'm going to feed into immediately to answer some questions. So you're basically putting it from start to an end. What is the endpoint of the knowledge that I'm building? So this is something which is an important thing. Second lesson. Better prepare a lot of training materials if you are out for a crowdsourcing expertise. We had to do a lot of one-on-one sessions with a lot of people, trying to explain them, where we are going with this curation effort, what exact intricacies of the curation we would like to have in mind, how annotations should be placed, and so on and so forth. So we are still lacking these training materials. So this is something we really need to think about developing, but of course you cannot do everything. So this is something that we felt really hard. That if there would be some kind of a set of prepared videos, just go watch the video and let's talk about it afterwards, or follow a hands-on session, training session, how to use, I don't know, Cell Designer or Newt or something else. That would be very, very, very helpful. And the third, like I said, there are so many. For the crowdsourcing or for the project this size, be transparent as much as possible about who does what and give credit at every possible step. In that type of projects, community projects, it's very challenging to drive it such that no one feels left aside. So engagement of people, or having an idea on how to engage people, is very, very important. And from the very beginning, you need to think about putting everyone in the places where they are doing something, and involving as many people as possible. Those would be the three that I can give you from the top of my head.
And very nice.
Yeah, so a disease map, a new disease map project could be accelerated by this effort. Yeah, I think so. Just from the point of view of all the automation we've done on the workflow, format translation, linking to text mining, looking for optimizing curation. I think this is there are a lot of things that we've learned. Yes.
So a comment from Alan James, super talk Marek, thank you for clear presentation. And one of the last question from Ahmed, thank you for the great talk. We need COVID-19 disease map, submap for the mechanisms of action of the mRNA vaccines?
Okay, that's a good question because I don't have an answer. I would instinctively say yes. Immediately, I started to think whether we can represent the mRNA vaccine mechanisms in the diagrams which we already have, or would we need something else? Because normally mRNA vaccine is, as far as I understand, it's producing viral components in order to stimulate the immune system. Hard for me to say. Like I said, a very good question. I don't know how exactly how mRNA vaccines work, to be honest. I know the general idea that you deliver the viral payload into the cell. You let the cell produce the empty virus, and then this stimulates the immune system. So that I understand. What is exactly happening with what is specific to mRNA vaccine, and what is specific to the handling of this incomplete viral payload, I don't know. Perhaps indeed we would need a dedicated set of mechanisms to represent that. So I would say maybe yes, but it would be interesting to see if we can represent this just using the diagrams we already have.
So I think there are no more questions, unless, I don't know, you want to mention something at the last comment or something, or there is, I don't know, I'm just saying, okay, there is one more. But I think this one from us is already addressed. So the one with the variant-specific knowledge, like putative molecular basis of different responses of different various variants to immune system and drug challenges. So this was already addressed also.
Yeah, the only thing I would like to comment here to the question of Thomas is that we partially are exploring this by this multi-omics visualization I was showing for different cell types. If you show them side by side, you have an insight into cell specificity mechanisms. It was possible to be seen that these airway secretory cells 3 are the only ones which pop up at the virus replication cycle. For the variants or for any, you could say, tissue or system-specific or cell type-specific questions, you could try answering them this way, putting things side to side, but just simple visualization, putting them side to side and observing the differences. I don't know from the top of my head how I would employ this analytically, but I think it still should be possible. So either you map this on side by side from the point of view of different data sets, or you provide for variants, you need to have a mechanistic explanation. Because I understand that the variant might be associated with the disease, but as long as we don't have any mechanism, we cannot act on it. If the variant is very much segregating between, say, susceptible and not susceptible, if it falls in a non-coding region or a promoter region, it's very difficult then to, without a careful examination of how it affects the transcription and of what, it's very difficult to go into that without this information, so without mechanistic interpretation.
Okay. And then a comment from Nuria. Impressive community effort and great talk. Thank you, Marek. Have you worked on how to integrate this disease map information with patient clinical information to add the clinical practice? Thank you again.
Yeah, thanks a lot, Nuria. That's a great question. It's doable. I would say it might be doable in the same way as we discussed for cell specificity. But for that, we really would need to have access to patient-specific data or subgroup-specific data, perhaps calculate molecular profiles which are specific to these subgroups and then put them on the map and try to visually analyze them, or to focus on the molecules or expression profiles which are differentiating between the groups, and then rerun the analytical pipelines for those to see what might be behind this. I think that in the end, the computational workflow might be quite similar. You calculate differences between the groups, and then you try to make sense out of it in the context of the map. But getting access to that is another pair of shoes, and I think it might be quite challenging. So what we were presenting in the map so far, we relied on the generosity of people who have already did the experiments and provided the publicly available data sets. For the patients, yeah, I don't think it would be as straightforward. Would require some kind of a collaboration with a clinical partner who has a COVID-19 cohort and has molecular readouts, which then we could try to make sense out of. I think in general it's doable, but requires all the GDPR related or sensitive data related work out first.